My name is Atis. I am as a postdoc in the Jonathan Watts lab. I want to thank you to the OTS webinar organizer for giving me this opportunity to share my work. I'm truly honored to present my part of this work in a series. Too excited to discuss our recent advance in the RNA purifications. Just we can, yeah, sorry. Oh, I cannot see my presentation.
Oh, we can see your main screen. We can see your cursor.
Stop and share again. Yeah.
Now we can see the PowerPoint, the whole program.
Now it's
now we see just the slides.
Yeah. The title of my presentation is Enhance the Purity of sgRNA and pegRNA by using the piperam terminal dioctyloxytrityl protecting group. Overview of my presentation: importance of RNA purity in therapeutic application, challenges in synthesis and purifying long nucleotides, dot modification as a solution, key advantage as a dot purification, and conclusion and future applications. In the introduction, I just want to tell you why purity is important. Purity is important is a paramount ensuring the safety, efficacy, and regulatory compliance. Impurity can lead to adverse effect, reduce potency, compromise overall the outcomes. Impurity can impact on pharmacokinetics and pharmacodynamics. High purity compound minimize the risk of toxicity and allergic reactions. In the field of oligonucleotide, the synthesis and purification of CRISPR guide present a significant challenge. If the synthetic challenge, if you see this graph, when the stepwise yield, if you look the stepwise yield, when oligonucleotide drops very sharply unless the stepwise yield exceeding high. If you saw, this is the like 99%, and then it is a good yield, but other, if you go to like 99, it's very low yield. I want to highlight here in the case of the guides, where is the single guides and pegRNA is exceeding than the 100 mers. If you look the 100 mer stepwise yield, if it is a 99.5, it's like a theoretical type yield. We will generally get 98 to 99% yield in between. Like if you saw suppose we start with the 1 micromole synthesis, we'll get like 130 nanomole to 260 nanomole type, like 13% to 36%, 35% yield. And another thing is as a similar as like longer is harder for purification because of if you look the percentage difference between n to n-1 species, when this is a ASO, if you consider percentage different from 1 n- full length to n-1 product, is like nearly 6%. If you case of the sRNA is nearly 5%. But if you look in the single guides, the percentage difference between like 1%, and then it's very difficult to purify. It's similar like a pegRNA is nearly by 150 mers, and the difference between less than 1%, it's very harder to purify. What is the current method for purification for this long mers purification? People using like fluorous tagging, but it's very high cost, synthetic complexity, because it's very lot of fluoride atoms there, and this environmental concern, it can reduce the yield of overall compound and requirement of the special condition for the deep protection. Another, we can purify by purification via extended method. It take more time, and buffer consumption is take a more method, and when it's a longer one, you will get less yield also. And then another strategy is a DMT-on strategy, but DMT-on strategy is suboptimal for the long oligonucleotide. Why it is, I will show you in the next slide. If you look the single guide structure, in this single guide structure, it's very complex structure. When you as a DMT on the single guide, if you look, this is a very small. If I highlight this one, it's if I highlight this one, it's looking like this. Overall, this molecule, if you add the base here, is like a one nucleotide and this two CS3 and three benzene ring. It's not sufficient to give the hydrophobicity to purify like this 100-mer type compound. For this reason, for this one, I can show you when the LCMS spectra for the crude, when I synthesize the poly-T 100-mer, it is a very simple synthesis in the synthesis case when you have the thymidine. And then if you see, this is the impurity of this area. If I show you here, this is the peak. If you show like that much N-1, and this is the DMT on compound, still we cannot separate easily. It's still, this is the analytical column, but when it's actual synthesis, if you look, single guides, this is less than 1 nanomole on the analytical column. We cannot separate these two peaks from like impurity to DMT on. And then actual, when we add this suppose 1 micromole synthesis on the actual HPLC, we cannot see the DMT can separate from this whole group because the crude compound is more and we cannot separate them. For address this issue, we think about we can work on this R group, because this traditionally CS3 is the trityl group, and then we think about the different length of this alkyl chain. We think about the C4 increase the polarity, C6 slightly improve the hydrophobicity compared to C4. Then C8 is their option. There is a lot of options, but when we think about the solubility and purification, both parts, we think this C8 is a good choice because it increases the hydrophobicity also and solubility issue also solve. If you go more than this one, it maybe can reduce the solubility of the amidites, and then synthesis maybe synthesis complicate. Since we selected this one, C8, and then go for the synthesis. For starting the synthesis, we go the trialpha chlorine toluene with the octyl phenyl ether as a both starting material with the help of Lewis acid. Do the Friedel-Crafts alkylation reaction, and then we got this compound 3 is a dot alcohol, and then further this react with this acetyl chlorinating reagent with the presence of toluene for 6 hours, and we will get this compound 4. And this compound 4 we use as a dot chloride. Then I'm talk about more in my next talk like dioctyl chloride. I just mentioned the dot chloride. That means this is the structure of this one compound 4, and this compound 4 will react with the use as a 5' protecting group as here by using the DMAP and tripridine. For we use as 3 different bases and different chemistry on 2' position, just DNA and OMe also, and then we get the compound 6 with the drum base, and further we react with this in the 2-cyanoethyl NN dipropyl chlorophosphine is a phosphorylating reagent, and then we'll get this novel amidite. After this, if you saw the structure of this novel amidite, then if you can look, this is the increased alkyl chain, and we think this is useful for the purification of the long mers. And then we synthesize the different modified sugars and different chemistry with the compared to the DMT. Here in this case, just DNA poly-T, we synthesize the DMT on and dot on. Here is all modified sugars with the some PS is there, some modified 82% modified sugar, 12 PS is there, different this diverse synthesis we've done, and then we check the calculated mass and observed mass is similar. Just previous slide I showed you like purification of DMT on and dot on. If you compare this one with this dot on purification, if you saw this one is very retention time is more on the column, and then we'll get the more purity as compared to this one. And then we go to actual single guide synthesis. If you saw here, the DMT on peak it's very low scale. It's not 1 micromole. I just want to mention that one. When you have the low scale, we can separate a little bit this one. But if you saw this DMT on an impurity or OH product, it's separated from each other. But if you look the dot on, it's like too much separation from the N minus impurity, and you will get the high purity. If you look disadvantage of this one, is when DMT on is there, it's very close and poor impurity outcome. And then if you are dot on, your separation is more and retention time on the column is more, hence you got the higher yield also. This graph I want to show you, highlight one things more, the reactivity. If you saw this, the reactivity of the dot on and DMT on, if you saw the impurity, because impurity can tell us like how much reacted, and the pickup area also will tell. The both compared to DMT on and dot on has a similar impurity, means the comparatively DMT and dot reactivity is similar. Then comparative analysis for the DMT and dot on deprotection after we purify this one and how we will deprotect that one. The method for the DMT on guide to deprotect, we use the 50 millimole TEA buffer with the pH 5 and then 40 degree centigrade for 60 minutes. Just keep that. After heating, we will get the 5' OH product. Similarly, we tried this one with the dot on, and it shows the similar effect with increasing the time as 60 minutes, and it here require the 100 minutes, and we will get the similar quality of the full length product. We have done some different example for this one. Purified by dot chemistry is like 100 mer DNA, 99 mer single guide, 120 for pegmers, and then we got the good quality oligo for the long mers. After this one, the main advantage of this one is the yield recovered. After purification, how much yield recover? With this graph is related to 1 micromole guide synthesized guide for purification. If you just use the without DMT on, we'll get from 1 micromole single guide nearly 10 to 12 nanomole pure compound. But when we do the DMT on, it will increase like 15 to 20 nanomole pure compound. But if you use the 5' dot, you will get the more compound, like 25 to 30 nanomole pure compound after purification. Key advantage of this dot purification: shorter purification time, higher efficiency for the long oligonucleotide, compatible with the existing method, no special reagent. Without this dot modification, we just require this modified amidite, and then we can use anywhere. Conclusion for this work: the traditional purification techniques struggle with the challenges of the longer oligonucleotide purification. The 5' dot on modification offers significant improvement. This method streamline in the process, reduce time, produce purer product with higher yield, making a superior alternative for the oligonucleotide purification. At the end, I want to acknowledge my lab mate and firstly, I want to acknowledge my Professor Jonathan Watts for his invaluable guidelines and support throughout this work, my lab mate for their collaboration and daily insight, the RTI department for providing essential technique and support, and UMass Chan Medical School for the research environment and researchers that made this work possible. Thank you so much for attending.
That was great. I'll just remind everyone to put your questions in the Q&A. If you have questions, Ritesh, you can put them there. And even if you think of them while another speaker is talking, you can add them and we can answer them offline. So I have a few questions to get things started. I know that anyone who's making oligonucleotides, it's a challenge, especially when you're starting on a micromole scale and you're getting like, out of the micromole, it can be really like inefficient. And so I'm wondering, in terms of, other than this dot modification, have you learned any other like general tips that you can share with people about the like just getting higher yields in synthesis?
Other than the dot, we other than the dot, we didn't done, and it's other than this DMT on modification, there is no modification known in the literature. I think so, my knowledge. The I never heard people tried the fluoros tag also on the single guides because it's very complicated. I don't think so. Other than this one, in my knowledge, there is the another modification can use as a tag on the pipe position.
And what about in terms of like just preparation or downstream working with the sequences? Do you find it's better, like, you know, if you have a humidifier in the room, or like, because with our synthesizer, we have a whole bunch of little tricks like that we use to try to increase yield. And so I'm just wondering if there's any tricks of the trade you have to share.
Increase the yield other than this dot. Sorry, I didn't get the question. Can you just repeat one time, please?
I was just asking, so for example, with our synthesizer, we have to run a dehumidifier in the room to make sure it's completely dry. Like otherwise our yield is really poor if it's too humid versus, so we always say like, oh, make all your DNA in the winter. I'm just wondering if you have any extra tricks for people who might also be interested in using this dot modification, but they want to get the best yield possible.
Yeah, that's very good. You are using that checking the humidity of this room and then how much dry condition you can make. I can give you one insight. Like generally we are using the for drying the compound. If you want to synthesize your start the oligo before before making the before starting the synthesizer, you can make your amidite before like 2-3 hours and keep dry pack there, and make sure before starting your synthesis your solution amidite solution should maybe dried, because generally what we will do, like we will make the amidite and go on synthesizer, and initial yield is very important. Initial 10 mark 20 mark is very important to go long marks, because initial yield is a drop, then your long synthesis is pale. Pale means more time single guide and pegRNA is really challenging if you have
Okay, that's a great tip. Thank you. So we have some questions now from the audience. So one question is, are there any functional concerns with sgRNAs that have DMT on? Any toxicity concerns?
We are after the synthesis we are removing the DMT. I don't think so. That is the part of synthesis. When you are using on the synthesizer, you are removing DMT there, but we are removing after purification. I don't think so that can lead any toxicity, because after purification, after removing of DMT, you just amicon that one, your DMT will go away and your oligo will stuck on your amicon tube, and then there is no more DMT. I don't think so any toxicity because of DMT on conditions.
How does this method compare to TBDMS chemistry?
We are using TBDMS chemistry only for this synthesis for long modes also. If we have the other chemistry to available amidites, that's good for synthesis, because when you have the TBDMS chemistry, it's a little bit challenging for the long mark, but still it's working for us. So depend on synthesizer, lot of things matters. If you have the like inline synthesizer, it will work.
Okay, great. And then our last question before we move on to our next speaker is there a possibility of solvent fractionation-based purification or enrichment for large-scale synthesis with this hydrophobic modification?
Yeah, for the large scale, it is really useful. In future, we are thinking about like just using this one, just make a precipitate with some solvent and take the pure compound and remaining compound wash with this some solvent. It is useful for the large scale also.
Well, thank you so much. That was a great talk, and thank you for all your tips. I know synthesis is always a challenge, improving yield is always a challenge, so it's great to see the research you're doing towards that end. So our next speaker then is Svenja.
Yeah, hi.
Let you get yourself set up there.
So, can you see it?
Yep,
wonderful.
That's great.
Okay, great. Then also hello from my side. So my name is Svenja, and I'm part of the Hartmann Lab in Bonn. And I'm very glad that you all found the time to listen to my talk today, where I present what I had on my poster for the last OTS meeting. And what we're doing is basically we are developing a new principle or a new concept to adjuvant mRNA vaccines. And for that, we're using RIG-I ligands that are directly attached to the mRNA. And I'm going to spend some time on explaining the principle behind that and the immunology and yeah, what we're aiming with this approach. So we all know that an mRNA vaccine should be highly translated, so we get a good antigen expression and also presentation on the surface, so we can activate specific T cells against this antigen that we're introducing into the cells. And then on the other side, we don't want this mRNA to be sensed by the innate immune system, because a high activation of the innate immune system can lead to high interferon signaling and then translational shutdown as part of the antiviral defense. And one key player of this innate immune system is RIG-I. So RIG-I is a cytosolic pattern recognition receptor that senses 5'3" double-stranded RNA and leads to this interference signaling, for example. However, a little bit of this interference signaling can be very useful, because we also we not only react with an antiviral defense, but also with an upregulation of MHC proteins and also the secretion of co-stimulatory cytokines that can attract B and T cells, for example. So we want not too much interference signaling to still keep a good translation, but a little bit to upregulate our presentation and the co-stimulation. And the way we're doing this is that we have RIG-I ligands attached to the mRNA, and they only signal if they are really in close proximity to each other, which is why they only signal when they're attached to the mRNA. And we want to have this, want to hit the sweet spot where we have like just the right amount of interference signaling by having several modules for the binding of those RIG-I ligands, and by the number of those ligands we can control the amount of interference signaling, so that we get still a good translation, but also a little bit of interferon signaling to upregulate the presentation of our translated antigen, and also secrete co-stimulatory cytokines, which can then lead to a better B and T cell activation against our antigen that's produced in the same cell. And so we get an antigen-specific humoral and cellular immunity against the respected antigen, and then also protection against the virus that we took those antigens from. And in this approach, we decided to look at a model which has antigens from the MCMV, which is the murine cytomegalovirus, which is part of the herpes virus family. And so it is a double-stranded double-stranded DNA virus, And this virus has an envelope, so it has glycoproteins on the surface and in different combinations. And for this approach, we chose 3 of them that are supposed to build a trimer on the surface of the virus, and also one immediate early protein, which is a transcription factor, which is produced very early in case of infection. And so I'm going to use those names for the proteins that we encode on our mRNAs in the following experiments. So what I mean with that is always an mRNA encoding one of those proteins, if you see those names. Yes, so first of all, I'm going to explain shortly how we produce our mRNA vaccines. So we use IVT for that, so in vitro transcription, which I'm sure many of you are familiar with. So we use a plasmid template, which has the coding sequence for our antigen and a flag tag attached to it for the visualization of the translation. And it also has a 5' UTR and a 3' UTR. And to this 3' UTR, we can attach our ligands because it has several binding modules for our adjuvant reg ligands. We use co-transcriptional capping and N1 methylpseudouridine while we're transcribing our mRNA. And we use lithium chloride purification and HPLC to get rid of the byproducts that we might get during IVT. And then we get a very pure mRNA sample that we can then combine with our ligands, and then we can compare how the mRNA acts in terms of translation and immune stimulation with and without the ligands. So first of all, of course, we wanted to check if our antigens are actually expressed in the cells if we transfect them with the mRNA. And here we used HEK cells. So first of all, we checked with the mRNA without the ligands if we get a translation or an expression of our antigens at all. And you can see here very nicely. So in dark blue, we have the nuclei, and in light blue, we have a staining against the flag tag that is behind the protein that we express from our mRNA. So we can see a nice expression of all the 4 antigens or all the 4 different mRNAs. And here you can see only the channel where we have the flag expression. And what we can also nicely see is that we have the 3 glycoproteins mainly in the cytosol, which we would expect, and a transcription factor mainly in the nucleus. So this is very promising that we get a good expression when we only introduce the mRNA into our cells. And then, of course, the most important question was, what does our ligand do with the translation? Can we still get a good translation if we combine the mRNA with the ligands? So for that, we used FACS to quantify the flag staining. And as you can see here, we always have the mRNA and then the mRNA with the ligand with this red frame. And we can see for all of the different constructs that the ligand slightly reduces the translation. However, we still have a good amount of cells that express our antigens that can then be presented on the surface, if you compare it, for example, with the untransfected control. And what you can also see is what I call trimer here is just a combination of several mRNAs. So in this case, for the 3 parts of the trimer, because we wanted to combine the mRNAs for a multivalent vaccine. And here we just check what happens if we combine them, if we still get a good translation of each of them. And this is the case. So this is very nice. And then we get still a good translation with our ligands. And now, of course, the most important question is what can we do with our ligands in terms of immune stimulation? And for that, I just want to emphasize that it's very important that the mRNA itself does not stimulate the immune system, because this interference signaling by the mRNA itself is something that we cannot control. And we only want to add the controllable innate immune stimulation by adding our ligands. And we worked quite a lot of this, and for the sake of time, I'm not showing it here, but we're just finishing up the story and hopefully publish it soon. So if you have any questions for how to make this mRNA non-immunogenic, then you can maybe reach out or have a look out for the paper, hopefully soon. And yeah, so as you can see here, we did quite a good job. So the mRNA itself does not stimulate anymore. And we can see in THP1 cells, which is a human monocyte cell line, that they react with an interferon signaling if we add the ligand to our mRNA constructs. So you can always see the ones with the red frame are the ones with the ligand, and the one without is the one that the mRNA without the ligand that doesn't result in interferon signaling. And what is also nice is that we can see this also in murine BM disease, so bone marrow dendritic cells. And we can see the same, that the mRNA itself does not stimulate by itself in terms of interferon secretion. And then if we add the ligand, we can nicely increase the interferon signaling in those murine BMDCs. And maybe just 2 words on the controls. So you can see IBT4 here, which is a different RIG-I ligand, which also signals when it's just introduced into the cells by itself. And it's used as a positive control, and CA21 is used as a negative control. So it's also an RNA that shouldn't result in immune stimulation. And now, of course, the most important thing is if this is interferon signaling that we can control. And for that, what I did here is I picked one of the RNAs, in this case encoding GL, so one of the glycoproteins encoded on the mRNAs. And I mixed this mRNA, or I combined this mRNA with 0, 1, 2, 3, or 4 ligands. And by that, I can very nicely control the immune, the interferon signal that results from the transfection. So as you can see here, if I don't attach any ligands, or if I only attach one ligand, we don't get interferon signaling. And this is something we would expect because, as I mentioned before, they only signal, or they only can activate RIG-I if they're in close proximity to each other. So one ligand attached to the mRNA shouldn't do much, but starting from 2, we get this nice dosable interference signaling. And then, of course, we want to know if this interference signaling can also result in a better presentation of our antigens. And for that, we need the upregulation of MHC proteins and also the co-stimulatory factors. So this is something we also looked at. Here again, you can see the interference signaling of murine BMDCs. And here in gray, you can see the mRNA signaling alone. And then in black, you can see how the DCs react with interference signaling if we attach the RIG-I ligands to the mRNA. And here we added another control, which is HTDNA, which is also sensed in the cytosol, and it's independent from the RIG-I pathway. This gets important in a second. But what we wanted to look at is not only the interferon that is secreted by the DCs that we transfect with our vaccines, but also if the DCs are activated and produce co-stimulatory signals. And for that, we can look at the surface markers of those DCs, also with flow cytometry. And you can see here very nicely that we get a good upregulation of CD86, for example, which is one of the activation markers of the DCs. And even more important, the same is true for MHC proteins. So especially for MHC1, you can also nicely see that if we attach the ligand to the mRNA, so if we compare mRNA without the ligand with the one with the ligand, we get a nice increase of MHC protein expression on the surface of those DCs. So for MHC2, this is not super prominent, but this is probably because DCs already have a very high expression of MHC2 proteins. And then probably we can't change that much more. But the most important thing here is the MHC1 upregulation, and those can then better present the antigen that is produced within those exact cells because they are transfected, because they were transfected with our mRNA vaccines. And then obviously lead to T-cell activation that recognize exactly this antigen and can lead to specific immunity. So what's also important is that this effect comes from, so it's important to check that this effect comes from our ligands and not something else. And for that, we looked at DCs from RIG-I knockout mice. So they don't have a RIG-I receptor, And we can see that if we add the ligand to the mRNA, we don't get any increased interference signaling. So this is completely RIG-I dependent, what we see here. So it's basically coming from our adjuvant ligand that we attach to the mRNA. And the same is true for the activation markers or the MHC1. So we can really nicely show that our increase in MHC1 and activation markers comes from the adjuvant ligands. So this already brings me to the end. So I want to emphasize again that mRNA vaccines by themselves should be non-immunogenic, that we can really add controlled adjuvant activity and not get a too high interferon response, which leads to translational shutdown. And this is exactly what we achieved with our RIG-I ligands as adjuvants, because they can upregulate co-stimulatory factors and MHC proteins in a controllable manner, and then boost the B and T cell activation and against the specific antigen that we encode on our mRNA. And what we want to do now is we want to vaccinate mice with mRNAs with and without the ligands construct. And we want to see if this interference signaling in vivo also results in better T cell activation, for example, and how the mice react to our new vaccines. And in the end, we want to do an infection challenge. So we want to see if mice vaccinated with the mRNA with the ligand have a better chance of survival than the ones only vaccinated with mRNAs without an adjuvant. So that was it, and I thank you for your attention. I thank my group, and especially Jan and Christine, who worked together with me on this project, and also Luca Chichinshine and Maeva, and Gracian and Ivan, who are going to help us with the mouse experiments, and of course the OTS for giving me this opportunity. Yeah, and if you have any questions, feel free to reach out.
Yes, thank you, Svenja, for this excellent presentation. So also here, if you have a question for Svenja, please use the Q&A function. But obviously, I can start. So let's start with the different ligands, right? So I think you had 3 types of ligands. I think they are called GO, GH, and GL. And I think for the dose response, or not the dose response, but for the multiple ligands that you add, you only show the data for the GL. I think it's the right lower graph.
That's right.
So why did you decide to take this one? And do you also have the data for the other ligands? And how does it look?
So what's different here is not the ligands. What's different here is the encoded sequence on the mRNA. So basically, those are 4 different mRNAs. One codes for GO, one for GL, one for GH, and one for IE1. And those are different proteins from the MCMV virus. And those are just encoded in the mRNA. So what differs here is only the coding region. And in the end, we wanted to combine those different mRNAs to get an expression of different proteins, and so we have a better chance of creating specific T cells. And this, I chose one of those mRNAs, and then I combined them with different ratios of the ligand. So they're not different, they're just in different ratios. So I can attach either 0 ligands like here, or 1, 2, 3, or 4 ligands to the mRNA to control the interference signaling. So this is what I compare here. And I didn't do that for all of the mRNAs because they don't differ in the ligand binding site, only in the coding sequence.
Okay, thanks a lot for clarifying this. Also, again, probably it's the next slide. You look at the upregulation of MHC1, and you kind of explained also that there is a difference in response with MHC2. So can you explain the difference in response? So why is this reacting better on MHC1?
So DCs have in general a very high expression of MHC1 proteins, and I normalize this data to the negative control, but of course this negative control is already very high. So if you compare the controls here, we can't do that much if we add a RIG-I ligand in general. So I think what we're seeing here is that we already have a very high base level expression, so we can't regulate that much more with our ligands. But MHC1 can be nicely upregulated, and this is the important one for the T cell activation, because this is the one that presents the antigens that are within the cell or produced within the cell, including the one from the vaccine. Does that make it clear?
No, thanks. Yeah, I think it's clear. Question about non-immunogenic mRNA. So I think because this is obviously an excellent control and probably also a big challenge. So can you explain short how you did this? How do you make the mRNA non-immunogenic?
So there were several steps while producing the mRNA that we had to adjust. So what we did is we varied the coding sequence. So we altered the codons a little bit, but not the amino acids. And what we also did is we optimized the poly-A tail. But yeah, as I said, we're doing that in a very comprehensive manner now to maybe publish it soon. So I can't say too much about it, but yeah, what also makes a big difference is, of course, the HPLC purification, because often you get a lot of byproducts when you produce mRNAs via IVT.
Cool. Let's go to the Q&A. Question from Marc Antoine. So could you hybridize the RIG-I ligands to the poly-A tail instead of adding specific binding sites? It would reduce the size of the mRNA.
So basically, mRNAs always have a 3' UTR, and it's actually beneficial for translation if they have a UTR, and not only the poly-A tail after the coding sequence. So we wouldn't gain that much advantage by this, I think. And in principle, it would be possible because you just hybridize the ligands with a complementary sequence. And if you use a poly-T, it would be possible to hybridize to the poly-A, but AT pairing is not as stable as pairing with, for example, G and C. So I think it's better to have it on the 3' UTR, where you have also some Gs and Cs that pair to get a more stable attachment of the ligands.
Well, thank you very much. And I think it's time to move to the next speaker.
Great. So our next speaker is Faith.
Hi, everyone. Thank you very much for this opportunity to share my research that was presented at the OTS conference. I am Faith Kivunga, a student at the University of Bordeaux-INSAM under supervision of Professor Philippe Barthélemy. So my poster's title was Lipid-stapled oligonucleotide supramolecular structures as baits for anti-cancer therapeutic application. So the oligonucleotide that I'll be mainly focusing on today is the G-quadruplexes. And these are non-canonical DNA or RNA structures present in G-rich, guanine-rich sequences. And how they are formed is as a result of a stretch of guanines, where the guanine interact with another guanine through the oxygen hydrogen bonding to form a G-quartet that is mainly stabilized by cations, monovalent cations. So when these G-quartets stuck through pi interactions, they form a G-quadruplex. This G-quadruplex can fold into different topologies. depending on the strand direction and the lateral loops, and also the position of the sugar with regards to the guanine base. So with the parallel conformation, we have all the strands moving in the same direction, as indicated by the blue arrows, which is a parallel conformation and has propeller loops that join one guanine from one tetrad to a different tetrad, G-tetrad. And then we have another topology that is the hybrid, which is it's a mix of parallel and anti-parallel, where we have 3 strands in the parallel direction and 1 in the anti-parallel direction. And it is characterized by lateral loops, which are loops that join guanines within the same tetrad. And then we have the third topology, which is anti-parallel topology that is characterized by 2 strands in the parallel direction, while the other 2 are in the opposing direction. And they can have the lateral loops joining a guanine to another guanine in the same tetrad, or diagonal loop joining a guanine that is opposite to the other. And this can result in either the basket conformation, where we have the diagonal loop, and where we have only the lateral loop, we have the chair conformation. So these structures have been shown to be present in most of the oncogene promoters and in untranslated regions of mRNAs, and they act as regulators of transcription. And this is because they do bind to, they do allow transcription, they act as a binding site for transcription factors, where these transcription factors, like the MYC-associated zinc finger protein or the heterogeneous ribonucleoprotein A1, among others, bind to them, unwind them to facilitate the expression of the oncogene. In the presence of the G-quadruplex, the RNA polymerase is not able to transcribe the gene, so their function is mainly to unwind this structure for the expression of the oncogene. So when we have decoys or baits, which refer to the oligonucleotide mimics of these G-quadruplex structures that are present at the promoters of this oncogene, they tend to compete for the binding of the transcription factors with the structure present at the promoter, and this helps in downregulation of the gene through the these oligonucleotides sequestering the transcription factors. So a case in point is the KRAS promoter. It has a nuclear hypersensitive element, a region that contains a number of G runs, as shown by the 32R and 21R. And these have been shown to fold into a parallel topology. So decoys designed from this sequence have also been shown to downregulate the expression of KRAS when introduced into cancer cells. However, the main challenge arises in their stability in the topology, changing of the topology, because the main topology at the KRAS promoter and other oncogenes mostly is the parallel topology, and also their ability to be internalized into the cell, in that they need transfection reagent. So in this case, we are looking at how to stabilize these G4 decoys, how to control the topology into the desired one that will enable the downregulation of the gene, and then to facilitate the cell delivery into the cells, and also to try and come up with other novel sequences that are shorter to see if they have a far much better activity compared to the decoys obtained directly from the native sequence on the promoter. So to do this, lipid conjugation has been shown to be able to stabilize these G-rich sequences into a parallel topology. And this, once this once these G-quadruplexes are formed, they can self-assemble thanks to the lipid to form nano-objects. So with that, we want to utilize the lipid conjugation to stabilize our G-quadruplexes, to make them to have a parallel topology and to also facilitate cell delivery. So solving 3 problems using one, with one solution. So in the illustration, I'm showing the formed G-quadruplexes that mostly for the short sequences, we form tetramolecular G-quadruplexes that are that have a parallel topology, and when they self-assemble, they form nano-objects that are able to be internalized by the cell. And these nano-objects provide an array of decoys that can sequester the transcription factors and therefore downregulate the expression of the oncogenes. So with this, we designed short G-rich sequences, and then we conjugated them to a nucleolipid, and then compared the activity of these short G-rich sequences to that of the native sequence, that is KRAS 21R sequence, in the activity for downregulation. So first of all, we characterized the nano-objects formed as a result of lipid conjugation. We did prepare the oligonucleotides in physiological saline conditions as both high levels of potassium and sodium tend to stabilize the G-quadruplexes, and those are the kind of conditions that are present inside the cell and outside the cell. And we see with the lipid-21R as an example, we see the formation of nano-objects using DLS. And also when we resolve them in the agar-rest gel, we can see that the lipid-conjugated oligonucleotides tend to form distinct bands of heavier molecular weight as compared to the non-conjugated sequences. And we see the same with the transmission electron microscopy. So to validate the formation of the G-quadruplexes, we use circular dichroism, and at the physiological temperature, 37 degrees, we see that we are able to form G-quadruplexes with a parallel topology, mainly characterized by a negative peak at 240 nanometers and a positive peak at 260 nanometers. And for the oligonucleotides that were not conjugated, they're not able to form the G-quadruplexes. So this was again confirmed by theoflavin assay. Theoflavin is very flexible. However, in the presence of G-quadruplexes, they tend to bind into the groove and their flexibility is constrained, and therefore that makes them to fluorescence. And in the graph, we see the fluorescence is higher where we have the formation of G4 quadruplexes and mostly in the lipid conjugated oligonucleotides. So now that we are forming an objects and G-quadruplexes, are they really able to bind the transcription factors as we proposing? So with this, we use the recombinant unfolding protein 1, which is an N-terminal peptide of heterogeneous ribonuclear protein A1 that is known to bind to the KRAS promoter and unwind it. So we incubated our oligonucleotides with the recombinant protein UP1, and we ran the gel with oligonucleotides with or without the UP1 protein. And as we can see, with the lipid-modified oligonucleotides, we see retardation in mobility of the lipid-conjugated oligonucleotides interacting with the unfolding protein 1, lipid G42, lipid G43, and lipid KRAS 21R. So this proves that the transcription factors are able to bind our decoys that we will introduce into the cell. So this possibly explains the results that we obtain with our Western blots when we treat the cells with the oligonucleotides. and extract the proteins and run them on a gel. We see that with the lipid G42, there is decreased expression of the KRAS protein, and we also see the decrease in expression of the c-MYC proteins. And as these proteins are involved in the regulation of cell proliferation, survival, and differentiation, I think this better explains our results with the cell growth inhibition, as the downregulation of the oncogene results in the inhibition of the pancreatic cancer cells treated with the oligonucleotides, regardless of whether they are KRAS addicted or not. I hope up to this point I've been able to convince you that with lipid conjugation, we can form stable tetramolecular G-quadruplexes in short G-rich oligonucleotide sequences. And this and lipid conjugation also facilitates the formation of the G4 parallel topology present in most oncogenic promoters, and also improves the anti-cancer activities of oligonucleotides, as the self-assembly of the nano-object formed as a result of the self-assembly provide an array of baits in which they can trap the transcription factors and therefore knock down the expression of the oncogenes. So I wish to appreciate all my lab members that help with this project. The OTS for giving me an opportunity to present my research, and all the funders of this project. Thank you very much.
Yes, thank you very much for this also, again, excellent presentation. Again, if you have a question for Fate, please use the Q&A function. And I can always start with a question. So I think you make use of very short G-rich ASO sequences of only a few base pairs. So how can you make these specific for specific targets? Because I think you mentioned that the G-quadruplexes are known in the oncogene promoters, but they're also present in UTRs of mRNAs. So would you not? So I think the proof of concept is nice now, but are they not very unspecific?
Oh yes, these G-quadruplexes are quite unspecific, I can say, because we are observing the knockdown of quite a number of oncogenes. I think it's good news, but then I think in terms of formulation, there is a way we can improve the specificity through adding maybe some targeting moieties to the cells in which we would like this downregulation to occur.
Yeah, so I think so. Since you target many oncogenes, you also target the UTRs of regular proteins, right? Are these also not affected?
I can't tell, but what I would think is, if I target this G-quadruplex into a cancer cell, I think it doesn't matter whether it binds to the UTR or not, but the killing of the cell, I think, is the most important thing.
I fully agree on that. Throughout your experiments, I think you used in your assay you used quite a high concentration of your compound. It's like 10 micromolars. So are there ways where you can reduce this concentration? Because I think that might be slightly too high.
Yes, the concentration is truly high, and we are looking at different ways of formulation, in terms of delivery, to see if we could be able to reduce the concentration of these oligonucleotides, something we are working on.
Yeah. So also, are you planning to because I think most of the work that you did is the in vitro work. So are you also planning to move to in vivo works, and how would you do that?
well, there are plans to do the in vivo work. However, like with my time, it's quite limited for now, though it is possible to collaborate with people who have the mouse models with this kind of disease and see how we can downregulate the expression of the oncogenes. Yeah.
Cool. Thanks a lot. I think I give the word back to Erin.
Great. Well, that was a great talk. Thank you again to all of our speakers today. It was really great to see really kind of some different stories of what's going on with the trainees in our community. So I encourage you to share with your friends and colleagues, tell them that the webinars will be up on the website. You can also check out the webinars tab on the website to see what upcoming sessions there are. So we have some planned for June and later in the summer. So definitely take a look back. And as always, if you're interested in presenting, you can reach out to me or Ronald, or you can email the info at oligotherapeutic.org, and we can connect you and start talking about doing a webinar. So thank you again to all of our speakers, and I hope everyone has a great day.